My dear students, clear, correct and concise shall be the logo for the tips for MD Path students. And we shall go to the next class regarding the techniques. The topic for the day shall be cytospin and cell block. Either of these, either combinedly or as individual topics, have been asked as university questions. In the first class, we have dealt with liquid-based cytology, in which the material is collected, it is suspended and studied. In this case, there is an exfoliation of cells constantly taking place from the surfaces of the body. So it is basically based on exfoliative cells, which are again being gathered at a particular point by means of centrifugation. It can be either the R.A.T.S, the conventional centrifuge, or the Shandon, cytocentrifuge. The rates of rotation are given over here. It is variable according to the sample and the experience of the cytopathologist. The material is kept in alcohol or formalin for fixation, following which smears are made and it could be studied with H&E or PAP. Before I go into the cytospin or the cell block, let me tell you what are the different modalities of collection or the samples. They can be direct smears, or they can be sediment smears from the urinary sediment you are making. They can be centrifuge smears. Sometimes they can be filtered and from which you are preparing the smears. Cell blocks can be made out of the remnants of the material. And the cell block itself can be used for making thin sections. The main idea of this is to improve the cell yield, the cytospin. The main purpose is to improve the cell yield. And what are the factors that determine the cell yield? It will be the mode of collection, first and foremost. If it is going to be admixed with blood, naturally our material will not be satisfactory. Fixation, otherwise there will be artifacts. Sometimes you find that fluids are collected, left overnight in the ward or theatre and sent to the pathology department the next morning. Preservation before processing. Obviously, there will be artifacts and autolysis would have set in. Therefore, preservation is important. If it is not possible, at least it should be refrigerated. Then, finally, proper preparation and staining, mounting and reporting. As I mentioned earlier, this list, it will haunt us repeatedly, but we can memorize and reproduce. Direct smears, centrifuge smears, cell block, and what is the cytospin? This is our main target for the day. And the different fluids that could be sent to us for this can be any of the cavity fluids, pleural fluid, peritoneal fluid, ascitic fluid, CSF, urine, and hemorrhagic aspirates also. If it is going to be non-hemorrhagic, you treat it with EDTA, and if it is going to be hemorrhagic, anticoagulants such as heparin can be used so as to prevent fibrinization or any clot. And how do you people collect the samples? They could be collected directly onto the glass slides, made smears of. Simple centrifugation. The R.A.T.S centrifuge that I mentioned. Cytocentrifugation. This I will be explaining in detail, and you people shall and will have to also do it. Preparation of the cell blocks. So please do remember this. As I mentioned earlier, this will haunt us repeatedly. The importance is, it is a fairly reliable procedure. It obviates exploratory surgery. Every time the surgeon will not be able to open up a cavity and obtain a biopsy. Cytospin, there is no overlap of cells. This we are also seen in the liquid-based cytology. Better cellular morphology. These are all some of the advantages or the importance. Further to this, fixation is simple, less expensive. Mesothelial cells, red cells, etc. can obscure the morphology, but this can be minimized in a case of a cytospin. If you are not impressed, look at this valve. What is this valve? Bronchio or bronchiolo-alveolar lavage of the fluids. And in this, I am able to see the random distribution of the cells. Distribution of the cells means from this end to this end, I am able to find the cells. Secondly, the cells do not overlap on each other too much. I can make out the cellular morphology. Always I used to tell the students, the difference between cytology and histopathology is, cytology is like preparing an egg omelette. You find that it is spread out, whereas histopathology is more like a cake or something. So here I find that the cells are well spread out. There is a nuclear detail, cytoplasmic detail, etc. What is lost is a pattern, but of course in fluids we do not have pattern. The advantages are mentioned here, monolayered. You can make out the reactive mesothelial cells in this. And sometimes there is what is called as the hypersegmented neutrophils. So this happens to be a case of respiratory distress syndrome. Of course, that was there for us in the last 1 year. And clarity and concentration. There are two sets of material. This is by means of the cytospin, and this is by means of a direct preparation. So in this first slide, I am able to see the cellular detail. They are being well separated, and nuclear cytoplasmic differentiation can be well brought out. The cellular yield is also high enough. This is in a lower magnification to show the quantum of cells. Same thing, if it is kept under a higher magnification, I will be able to appreciate the cellular details. Whereas here it is sparse and more thicker, I will not be able to make out the details as much as this. Rather, there is a chance that I could miss a finding. What are the principles of the cytospin? Now comes the major answer. Please memorize each and every word that is on the screen. The cytocentrifuge is a microprocessor controlled cell preparation that makes use of centrifugal forces to deposit the cells on the slide. It is too difficult, but then this word alone we shall remember. Centrifugal means tending to throw off the center. Centripetal means tending to draw towards the center. These are the two differences. Before going to the parts of the cytospin, let us look at the machine as such. I find that there is a facility for keeping the gadgets over here, and there is a reader and there is a lid. There is also a provision for auto locking, because if we do not lock it, the things will be flying to the roof. The entire gadget can be autoclaved because the samples can be positive for hepatitis, positive for HIV, and nowadays COVID. All the chambers can be disposable and they are provided with caps, which I shall be showing. Safety alarms are there to indicate the time if something is wrong, etc. And there is also a control panel by which we can operate from outside without opening it. So these are all the parts of the cytospin. Kindly do make a note of it. Try rehearsing it after I have said. This is an extremely important one. In some centers they are routinely using it. whereas in some others it will be just Greek and Latin. So this is a slide holder. There is a hole over here through which the material will come onto the slide. This is a filter, and if you look carefully, there is a hole in this also. Through this only you find that the material will be coming onto the slide. Now what happens is, this is being fitted in, and after that there is something called the funnel. I find a Y-shaped material that is actually a funnel. The fluid is poured into it, and once the centrifuge is switched on, you find that there is a deposit of the material which will be transmitted through this hole onto the slide. Also you find the filter paper absorbs all the unwanted material. This again is explained over here. So this is a funnel, this is a tube, and this is a filter. You find that there will be a hole, and through this it is transmitted onto a slide which is fixed onto the slide holder or a metal clip. So the principles are centrifugation and centrifugal principles tending to throw off the center. The cytospin can deposit the cells in a defined area. That again is an advantage. We need not be searching for cells throughout the slide. The residual fluid is absorbed onto the filter paper or card. There is a flattening of the cells, excellent nuclear presentation, and the cytofunnel. It helps in holding it upright for the equal distribution of the cells. So it will not be slanting or whatever it is. So there is a sedimentation, the cells pass through this, and it is getting deposited onto the slide. I don't think you can get a better diagram. Look at this again. It looks a little complex, but it is simple. So here there is a funnel. As I had mentioned earlier, there can be a lid for it. Once the centrifuge is on, nothing will be splashed. And this is the slide that will be marked. In this ring itself, you find that the cells will get deposited. Unwanted material will be absorbed by means of the filter paper, and this is a holder for it. So look at this one. For example, there is a slide that is fitted in. The funnel is also over here, and after it is fitted in, it is kept in the slot in the centrifuge. The process is done. So this particular picture, it looks very complex. In fact, it is a wonderful summary of the entire process. One word is, it has to be balanced. Once I put a material over here, exactly a similar volume should be kept on the opposite side. Otherwise, there will be an imbalance and the centrifuge will be dangling. The use of it can be, so this is actually supposed to be the stem cell. Stem cell is little complex. I used to think that it is somewhat weird and what, but it just looks like a large cell, like a histiocyte or a large lymphocyte, and it can be used for immunofluorescence. I am not going into the details of it, just to show how much the cytospin can be useful. Fluorescence binding HL60 cells. If you go to the net, you may be able to find this. And most from the least. Sometimes you find that the material will be sparse, but when there is a concentration, the pathologist is at ease to study the cells. And here not only the prominent nucleoli, but also some pattern of arrangement can be made out. So it is a case of a metastatic adenocarcinoma. Here again, amidst the mesothelial cells, there can be the foreign cells that could be made out. If there is any doubt, from a cell block, a cytochemistry, immunohistochemistry can be done. So this is the fluorescence that I had been talking about. The details are given here. Just see the keywords that are being highlighted and underlined. Fluorescence binding HL in a suspension. And also you find that this is fluorescent method to highlight the microtubules. How do you prepare the cell block now? Now that we have finished with the cytospin, what is the cell block? You can use any of the tubes. It can be a conical tube that is preferred. And there is a fluid. In fact, you can use a diluting fluid also, and there is a centrifugation. As I told you, always opposite side must be balanced. There is a sedimentation over here. It need not be at tremendous speed. Even at moderate speed, you find it will be there. The supernatant is decanted and the material is prepared. It is prepared out, and if necessary you will be able to add another material, or you can add plasma and thrombin. Plasma and thrombin, there are some controversies about it, which we shall be seeing. And finally, a cell block is prepared, just like tissue processing, and you fix it in formalin, the process goes on. This is not for rapid diagnosis, it takes a time as routine tissue processing. Height of innovation in a cell block using plasma and thrombin. See what is being done. So this is a graduated centrifuge tube. So in which after the spinning you find that there is a sedimentation which I can make out. The supernatant is decanted and they mix with plasma and thrombin. What they say is that it provides a fibrin mesh or a gel-like material which has a binding onto the cells, and finally a paraffin block can be prepared out of it. The problem with this is, we shall see in the next slide. It can be a potential source of DNA contamination. So when we do it like this, we are using the plasma and thrombin, the DNA actually it can be contaminated. So when we do subsequent studies, it might not be understood. So therefore we will have to pay the penalty for it. But anyway, it is a novel idea, have it in mind. Plenty amidst poverty. When I say poverty, it means paucity of the cells. And particularly when there is a stromal lesion, the cells will be few. Because of the cytospins, you get the collection which can be used for a James stain or a quick stain, for cytochemical stains, immunohistochemistry or immunocytochemistry, in situ hybridization, as well as laser microdissection and microarray testing. Each of this is a wonderful process, a world in its own, and for all this the cytospin can be made use of. That is why I called it as plenty amidst poverty. Look at the number of techniques it can be made use of. What are the factors determining? One is the method of collection, whether the fixation is apt, preservation of the fluid prior to processing, preparation of the material for staining and microscopic examination, and mounting the cell sample. Otherwise, you can just remember this. In ordinary centrifugation, there is insufficient material. A cell block can be prepared from the residual material also. This is regarding the CSF. Give unto others what you have. What does it mean? I am not taking it from the Bible, but see this. It is a CSF, it is a protein diagnostic material. You find that there is a centrifugation. Fluid is over here, it is centrifuged, and then there is a collection of cells. This material can be given for protein estimation to biochemistry, and with the rest of it I can provide a suspending medium, again recentrifuge, and the cell block or the cell buttons can be prepared, from which there can be a staining done. So this is just an outline of it. Look at this picture. It is a Shandon cytospin. Cytospin is a centrifuge that is used specifically for cytological purposes and preparation of a cell block. We should really appreciate the world of precision. Nowadays, most of the gadgets are here. Therefore, I do not know whether we are giving into science or the science is helping us. And from this, a cell block has been made, a papillary lesion could very well be made out. What are the advantages and the disadvantages of each? From a cell block, the advantages, the architecture is brought out, serial sections could be produced for immunohistochemistry, slides can be preserved, and thinner sections can be made. Whereas the disadvantages are, it is just like routine processing, time consuming. Morphology can be modified by the type of fixative that we use, and ample sediment might be required. Very frequently we say it is inadequate for diagnosis. Cytospin, on the contrary, you find that there is a better cell concentration even in hypocellular samples, better cell morphology. Artifacts are lessened, less time consuming, inexpensive, less manpower is needed. These are the advantages of cytospin. But the disadvantages are material like RBC, WBC, other debris can interfere. They can obscure the epithelial cells. Mesothelial cells can get aggregated. They will form all forms of mimics. They can even mimic an adenocarcinoma. false acini, rosettes, etc. can be formed. All this can be improved by using cell blocks. So the advantages and the disadvantages of the cytospin and the cell block, please by heart, it is important for you people. And in this class we had seen the definition, the samples, the methods, the stains, the applications, machine particularly, the procedure. This will be the key for the entire answer. How you are going to load it, prepare the slides and ultimately do the staining and compare the cytospin and the cell block. And there is a machine over here. In order to understand, I destroyed myself. This is actually a backup on another saying. The world will have to get saved from dangerous weapons only by creating more dangerous weapons. These are the words of Alfred Nobel, who was the inventor of the dynamite. Today dynamite is no longer harmful, but then the atom bomb and the hydrogen bomb, etc. are there. So in order to understand, I have destroyed myself.